millions of people in the world living with a silent killer hiding in their bloodstream. So this killer called as the Trypanosoma cruzi. So without wasting more time, let's go deeper into this disease. Assalamualaikum to Dr. Akmal and my fellow friends. I am Syazwani Binti Izham, and my other school member will talk more about the parasite called as the Trypanosoma cruzi or T. cruzi. For the first morphology is the trypanosoma, which is the infective stage. The characteristic of this trypanosoma is the spindle-like structure and have a jelly with the undulating membranes. The second morphology is the mastigot, which is the intracellular replicative form and it lack of the visible flagellate. The last one is epimastigot, which is the vector replicative form, the vector that brings this parasite called as the kissing bug. As you can see from the poster, this parasite was distributed throughout the South and Central of the America. This parasite was transmitted via the bite of the infected kissing bug that present in the feces of the bug. The second one is maternal transmissions. The third one is the blood transfusion, and the last one may be the organ transplant. When someone was infected by the T. cruzi parasite, there are two possible symptoms, which is the first one is acute phase, the short period incubation, then can cause romana or trachoma, and the second one is the chronic phase, which is a long period incubation that may cause the death.
The first current method that I am going to introduce to you is a molecular method called hemoculture, a parasite detection in blood samples. Based on T. cruzi genetic characteristics, it is classified into 6 DTUs and an emerging DTU called TC-BAC. DTU, aka discrete typing units, are the genetic subdivisions of Trypanosoma cruzi. Therefore, in order to identify T. cruzi, we need to find its DTU in the host tissue. This study demonstrates a flow cytometry test called human Chagas flow ATE IgG1, which is used in detecting DTUs for Chagas disease. Alicia et al. 2020 used the hemoculture method to isolate T. cruzi DTU from each patient. After they have been obtained, they were inactivated until the assay of human Chagas flow ATE IgG1.
The second method is a serological method, which is ELISA. ELISA, or enzyme-linked immunosorbent assays, is a laboratory technique that detects and quantifies specific proteins such as antibodies or antigens in a sample for diagnosis. According to the article, T. cruzi expresses a protein called TC323 that is unique to the parasite and not found in other organisms, which makes it an effective biomarker for diagnosing T. cruzi infections. As the SPH is used to analyze and verify the presence of T. cruzi antigens, which are RTCD3 and also RTCD6 in the blood samples of chronic Chagas disease people. Next, the antigens are used to coat the wells of the ELISA plate, followed by the addition of blood samples to the plate. If the patient is infected with T. cruzi, the blood samples should contain IgG antibodies that bind to the antigens. Furthermore, a secondary antibody that is linked to an enzyme is added and binds to the antibodies that are already bound to the antigens, which produces a color change. Then, a color-changing substrate, which is tetramethylbenzidine, is added and measured by the Versamax ELISA plate reader at 450 nanometer. The higher optical density indicates the presence of T. cruzi antibodies, which confirms the Chagas disease infection.
As you can see from the poster, hemoculture have a lot of disadvantages than the ELISA. As conclusion, ELISA is the best method used for diagnosing the T. cruzi because it's highly effective.